Good morning, everyone. Welcome to the PAG webinar series supported by Daicel Arbor Biosciences. Thank you for taking the time out of your day this morning to join us, and thank you to Daicel Arbor Biosciences for your partnership. Today's topic is sequencing solutions for agricultural microbiomes and pathogens. The question and answer portion of the webinar will be at the end of the presentation. You can submit questions via the Q&A function at any time throughout the event. Any questions not addressed live will be answered via email. We would love to hear your feedback, so please submit your questions and comments. I would now like to introduce our speakers. On today's webinar, we will have Dr. Brian Brunelle, Senior Product Manager at Daicel Arbor Biosciences. Since 2014, Brian has been the Senior Product Manager for MyBaits at Daicel Arbor. Prior to his role at Daicel Arbor, Brian was a research microbiologist studying multi-drug resistant salmonella in pigs at the USDA ARS National Animal Disease Center. He earned his PhD in infectious disease and immunity from UC Berkeley. After Brian, our next speaker will be Dr. Brook Anderson. Brook is an R&D scientist at Native Microbials, responsible for evaluating novel host-derived probiotics for animal health and productivity. She holds a PhD in biological sciences from UC San Diego, where she specialized in microbial ecology using model system cheese rinds, and a BS in biochemistry from the Ohio State University. All right, I would now like to turn this over to Brian to start the presentation. Go ahead, Brian.
All right, thank you. Thanks everyone for joining today. I'm Brian from Daicel Arbor Biosciences, and today I'll be talking about some NGS workflows for agriculturally relevant microbes. This talk is focused on practical decision-making, and the goal is to give you a practical framework for choosing the right approach based on your question, sample type. And before we get started, again, I'm currently a senior product manager for MyBaits at Arbor. Before that, I worked as a research microbiologist studying multi-drug resistant salmonella in pigs for about 10 years. I've also worked on some Rumen protozoal projects. As you can see, I got my hand in a cow. And so I'm pretty excited to learn more about what Brook's research later. And here's the roadmap for today. I'll start with a quick overview of Arbor. Then I'll walk through microbial NGS workflows used in agriculture. Second half, we'll focus on the best practices for project design, including targeted panel design, followed by a few case studies, and then, of course, conclusion. So at Arbor, we support microbial sequencing through both reagents and services. On the service side, we provide end-to-end support from extraction through bioinformatics for short and long-read projects. On the kit side, we offer custom and pre-designed catalog hybridization capture panels. The key theme is flexibility. You can mix and match workflows depending on whether you need broad discovery, targeted sensitivity, or genome level resolution. Just to note that everything we do is research use only, and we don't offer diagnostic testing. So, why NGS in ag microbiology is important? Because NGS is transforming agricultural microbial genetics. And it lets us characterize microbes directly from complex samples, whether it be soil, plants, or animals, without relying exclusively on culturing. Compared to traditional approaches, this improves speed, sensitivity, and resolution, and it opens up questions where you don't necessarily know what you're looking for ahead of time. NGS tools are important in the One Health framework. As they can characterize agricultural microbiomes, detect and identify animal or plant pathogens, resolve genomes at the strain level for outbreak tracking, understand host microbe environment interactions, and evaluate pathogen evolution, epidemiology, and zoonoses. There are 4 major workflows I'll talk about today. Metagenomic shotgun sequencing, or MSS, whole genome sequencing from isolates, targeted amplicon sequencing, and targeted hybridization capture panels. And you can see these are kind of the general workflows. The first one, shotgun sequencing, basically sequences everything in a sample. There are no enrichment steps. Whereas the other three approaches, whether it be culture, target amplicon, and HiveCap, have some level of enrichment at a different stage of the process between sample and sequencing. And this is just a workflow comparison. You can see that depending on which, oh no, you're still seeing slide 2? So if someone can't see the updated slides, please refresh your page. Okay, great, thank you. So sorry if you didn't see other ones, but this is a nice summary of the different workflows, looking at cultural-based whole genome sequencing, metagenomic sequencing, targeted amplicons, and targeted HiveCap. Each has a pro and a con, and kind of depending on your research question really will influence which approach or approaches is the best. So for sensitivity, PCR and culture are very, very good. But if you're trying to sequence a full genome, you wouldn't want to use PCR primers. You'd want to do full genome sequencing. But if you can't culture it, or it's difficult to culture, or it's like a BSL-3 organism, then doing some sort of enrichment or non-culture approach really is the best method. So I'm just going to kind of go over all of these. As I was saying, culture-based whole genome sequences is extremely valuable. It allows you to isolate strains, do functional analysis, and get clean genomes. But it's labor intensive, time consuming, and can be biased towards organisms that grow well in the lab. Many environmental agriculture relevant microbes may be missed if you're not putting in the right media. Shotgun metagenomic bypasses culturing and sequences total DNA and RNA directly from the sample. So in this slide, there's no graphic that you would need. It's just going over the relevance of the 4 approaches. So culture is very good because it's sensitive and can get you a clean genome for functional assay. Now we're talking about shotgun genomics, which is a second workflow you could do, which you can get everything in the sample, whether it's total DNA or RNA. This means you can capture non-culturable organisms and get a much broader picture of the community composition and function. It's powerful for discovery and surveillance, but it can be demanding. As we mentioned, there's lots of factors that go into it that could, affect the quality. There's inhibitors. You need to sequence very deeply if you have a very complex, sample, whether it's soil or fecal samples. Well, one of the highlights for those who can see the screen is what we call MAGs, metagenome or MAGs. They're metagenome assembled genomes. So from metagenomic or total sequencing, you can then use computationally intensive programs to try to assemble all the viral, bacterial, plasmid, really whatever is in your sample genomes. And it's very powerful, and again, does not require any like enrichment or anything that might potentially bias the sample. And this was an example of doing metagenomic sequencing for genome assembly, the paper that maybe you can see. So sorry about this figure. Teams doesn't like us today, but basically just breaking down what is the benefits and the pros and cons of amplicon sequencing. So amplicon sequencing targets specific markers. It could be 16S, it could be ITS, it could be AMR genes, it could be like a virulence gene. It's very, very specific. And it's very cost effective. It can scale very well. I've done many a 96-well plate using PCR in order to screen colonies, DNA extracted from colonies for certain representational markers. But the downside is bias. The primer choice influences what you detect. The resolution can be limited, and you don't get much direct functional information. It's great if you're monitoring and doing comparison tasks, but it's not ideal for genome level questions. And so another approach is hybridization capture, or HybCap. And HybCap works by using biotinylated probes to, fish out sequences of interest from an NGS library before sequencing. The advantage is efficiency. You can increase the fraction of reads that matter, especially for low abundant pathogens or functional genes in complex backgrounds. It's also more tolerant of sequence variation than PCR in many cases. The main trade-offs are upfront design considerations, ensuring you've tested the panel to make sure it works for your needs, the diversity you expect. And if you can see the pie graphs, the upper blue one with a little green is the amount of your target if you sequence the full sample, and then the lower one that's mostly green is your target if you do enrichment. It really prioritizes and gives you your target that you are interested in. And so here's a couple of comparisons between HybCap and amplicon. Which, yeah, PowerPoint is not working well for us today. But the general idea is amplicons come in pairs, they work together, they're spread throughout, and you can only use so many in a sample at a time, several hundred, maybe even 1,000 amplicon pairs. HybCap are independent probes that cover the entire region of interest, and it could be as small as 10 kb for some small viruses, or we can make a panel with millions of probes. For instance, so Haley, it really depends. You can go from 1% to 80%, but it also depends on the amount of background. If you have a lot more background, you'll get a less target, but there are ways to then improve the sensitivity. And there's a workflow coming up on that I'll discuss. Nope, no isolating. You could just extract DNA directly from an animal and look for the bacteria or soil or whatever sample you're looking at. And so yeah, HybCap can be very effective and work with very large samples. And so at Arbor, we implement hybrid capture through our product, our MyBaits kits. With MyBaits, we support custom and pre-designed targeted panels for microbes, including whole genomes and functional targets like AMR. We also have a microbial panel collection coming with pre-designed options for many ag-relevant pathogens. And for teams that want an end-to-end approach, we have our MyRead services that cover extraction through analysis on both short and long read platforms. Let's go back. And it's just an example. These data show the hybrid capture can achieve genome coverage comparable to just whole genome sequencing with far few total reads. This holds whether you're targeting a single pathogen or multiple pathogens in the same sample, making it especially attractive for complex ag samples. Oops, slide didn't go over. In this, we compare enrichment of respiratory pathogens compared to whole genome sequencing, and we get quite a bit of enrichment. And so for best practices for NGS projects, oh, this one doesn't have everything in it. Basically, I'm going to focus on 5 areas: the experimental design, the sample quality, targeted panel design, whether it's amplicon or HiveCap probes, and then the sequencing platform choice, plus data analysis. These are all tightly coupled decisions, not really independent steps that should be discussed as a single entity. So, general considerations. You want to define your experimental groups and variables up front. Sequencing depth should be based on your analysis goals. Ensure you have enough biological replicates to perform the required statistical analysis. You don't want to go through all that work and then have a reviewer say, well, you needed 3 more samples because you weren't able to do the correct statistical procedure. Plan your bioinformatics early so you can collect the right metadata and control and implement the right controls. And do consider controls. Negative controls can help identify contamination, and positive controls or spike-ins can help interpret non-detections, really verify that it was a a true negative, and are beneficial. And the controls are beneficial in making comparisons across runs. Extraction is often the biggest hurdle and a hidden variable that influences outcomes for microbial projects due to the nature of samples themselves. Extraction and storage method can dramatically alter community representation, so testing multiple kits is often worthwhile. When I was doing microbiome analysis from pigs, we would put the samples directly onto dry ice. Otherwise, we'd lose pretty much the entire anaerobic community. It might be worth test kits. We found that the Power Soil kit from, at the time, Mobio worked best on fecal samples for us, but people results may differ depending on the exact nature of their samples. And different ag samples contain different inhibitors. You could have humic acids in soil, phenolics in plants, biosalts in animal samples. And it's really finding an extraction method that works best with your sample. Library preparation is the next step. And it needs to match your molecule type and sample quality. Do you have high inhibitor burn-in? Do you have mixed taxa? Do you have GC bias? Do you have discordant abundances? There's really no all one-size-fits-all library prep. If you have degraded, if you're doing long insert, there's going to be considerations to choose the optimal kit for your needs. For targeted panel, when designing targeted panels, start with your biological questions. Are you looking for taxonomy, pathogen detection, functional genes, or whole genomes? Amplicons are sensitive to bias and divergence just by the nature of being a shorter probe, whereas HiveCap tolerates mismatches and degraded DNA. If you needed something that was allele specific for capture, you would have to use PCR primers. If you want something that would have variability that would allow you to capture something that might be 10% divergent, then you definitely would want to use hybridization capture probes. And always evaluate panels using mock communities or well-characterized samples to assess sensitivity and reproducibility. Bioinformatic analysis may need to account for high host background or background DNA and uneven microbial abundance that are common in ag samples. If most of what you're looking at might be, let's say, bovine, you might have to then do some in silico analysis to remove bovine reads. And you might want to try removing and not removing. There's definitely a lot of different approaches you could take depending, again, on what you're trying to accomplish. And so for HiveCap project planning, this is just a checklist. Oops, it was a checklist. You define the target scope. Is there going to be a host background or high copy repetitive regions that we need to avoid during the design process? What are the wet lab protocol choices? What sequencing platform you're using? And the downstream bioinformatics. Again, these decisions are all interconnected. Considerations when making a HiveCap panel, define the biological scope. Again, what are you trying to target? And the steps would be provide a target sequence We will do the bioinformatics for the design process, where we design overlapping probes, screen for problematic sequences, whether it's high copy, repetitive elements we want to avoid, host DNA, RNA that might be in the background, and then we provide the oligo sequences to you for approval. You keep the custom designs, but you're free to share them with anyone. We won't, but it's yours to control as you wish. And here's just a general workflow of the choices that could be made during the hybridization protocol. So you extract the DNA and RNA, you do the library prep, you amplify, you do hybs and washes. And this is where things can, there's more decision points. So a lot of times you could pool multiple libraries per capture reaction, which would save money. Typically people doing microbial analysis will pool 4, but you might be able to pool more samples, which would lower the price per sample. The big difference is like how degraded it is. Do you know the amount of endogenous sequence that are targeting? Because when you balance samples, you balance it based on target sequence, not just total mass. We do the Hive. Going into the Hive, input mass is important, should quantify for accuracy. We recommend at least 500 nanograms of library for short read or 250 nanograms of library for long read per library going into a capture. You could go up to 1 or 2 micrograms total per capture. You could actually probably go up to like 6 or 8, to be honest, if you have a high level of background and a small amount of endogenous target. And some applications benefit from even higher input. We do recommend at least 100 nanograms per library. We know people who will do like 10. It will work, but it won't give you all the data that you might want. And then there's a protocol selection. There's the standard approach. We have a high sensitivity approach. This is for the very rare targets. It could be short degraded, highly divergent. And then we also have a long insert if you wanted to use it with PacBio or Nanopore. So the kit is the same, but really all you have to change is the protocol. And the dual round capture is the high sensitivity. Basically, after you do an enrichment, you take that enriched library and enrich it again. And this really improves the specificity. This is very often used in the ancient DNA when they're trying to enrich bacteria from skeletons, where the majority of the sample is environmental bacteria, the next majority is human, and then a small fraction is actually the bacterial target. And sequencing platform choices. There is short read. Short read sequencing is accurate and cost effective, but struggles with repeats and assemblies. Long read sequencing excels at genome resolution, but requires more complex analysis and can be more expensive for data generation. And HiveCap works well with both. And so now to get into the data analysis and bioinformatics, again, bioinformatics should be planned up front. QC, host removal, coverage assessment are all critical. Depth of coverage, coverage uniformity and duplication rate, can inform downstream taxonomic or functional analysis. Here are some common workflows that people use. And please remember to document the version of each pipeline and database you're using. This will allow you to reproduce it. Even internally, if you get a different result, it might be due to the software, not anything else. All right, now I'll walk through a few cases where target capture really made a difference. These examples span pathogen genomics, virulence, gene profiling, AMR detection, and metagenomics, the latter of which you'll hear shortly from Brook. So in this study, they used hybrid capture to recover the first whole genome of Mycobacterium bovis directly from environmental samples. The data showed that environmental reservoirs are epidemiologically relevant, something that wasn't possible to demonstrate with traditional approaches. In plant pathology, hypercapture enabled target sequencing of effector genes that made up less than 1% of the genome. This allowed population scale analysis of virulence gene diversity that would be prohibitively expensive just sequencing the whole sample. All right, and this next example focuses on AMR detection, or antimicrobial resistance genes, where genes are often very low abundance. Capture reduced the total sequence reads by roughly an order of magnitude while improving detection breadth. More genes and classes of families were detected, including clinically important extended spectrum beta-lactamase genes. So the main takeaway is there's really no single best workflow, unfortunately. The right approach depends on your biological question, sample complexity, and resources. Targeted sequencing, especially hybrid capture, can help improve efficiency and interpretability for many ag microbial applications. If you want to talk through a specific project, please reach out. We are very happy to help. And thanks again for your time. And now I will, oops, I think there's a few old slides. I think this might be the old slides, not the new ones that were uploaded yesterday. So there might be a few.
Thank you so much, Brian. That was a great presentation. We really appreciate it. Yeah. You know what? We will now let's introduce Brooke. We'd like to introduce Brooke Anderson for her presentation at this point. Go ahead, Brooke. Hopefully we can get to your slides here.
Oops, sorry, Brooke.
Okay. Oh, no.
All right.
Yeah, we definitely accidentally, sorry, guys. We were trying to deal with some of the funny slides and we managed to make it worse right before starting this webinar. So I'll be missing some headers. Looks like we've got some formatting problems. So stick with me here. Definitely some slide issues. Maybe we can get you guys equipped with slides that are all fixed up after the webinar. My name is Brooke Anderson. I am a senior microbiome data scientist at a company called Microbials. I'll be showing you guys kind of what we've been doing to develop a novel probiotic for dairy cows to help them make more milk more efficiently. Wanted to give a bit of background about our company. Again, this is Native Microbials, founded just over 10 years ago in 2015. We're based out of San Diego, California. And our main priority has been to develop new microbial products for animals by sourcing microbes from the animal's own guts. The reason why we focus in on animals in particular, as opposed to something like humans, is partly because animals have this very homogeneous sort of genetic background relative to people. This is mostly due to breeding, right? And the livestock industry is a very perfect example of this, right? Since animals have been bred for very specific industrial traits that are relevant to that industry. Animals are also fed these scientifically formulated diets pretty much the same time every single day. And so a lot of the sort of environmental sources of variation that we see in microbiomes are reduced in animals because of this. And microbial products are not new to these animal industries. Livestock spends almost $1 billion a year on microbes, but they're all pretty much the same kind of microbes, like Saccharomyces cerevisiae, different kinds of lactobacillus or bacillus strains that were selected mostly because we are really good at growing them, right? These are byproducts of beer bread making, silage inoculation, yogurt, pickles, and they're not necessarily selected because they have a lot of relevance to the ecosystem that they're being fed back into. The dairy industry in particular is an industry where that has a lot of room for growth, and we think it's a good target for microbial products. This is partly because they're very invested in new technologies in order to improve milk production. So this picture on the left shows how milk production has been ramping up dramatically over the past decades, from 145 billion pounds of milk produced in 1985 to over 225 billion pounds being produced in the most recent years. And at the same time, the number of cows that are being farmed in order to make this amount of milk has dropped dramatically. And just from 2004 to 2024, there's been a 25% increase in the productivity of cows to produce the amount of milk to meet consumer demands. A lot of the reasons for these improvements have been due to sequencing-based technologies, especially improvements in breeding by using cow genetics. And so cows are now bred according to certain traits that they are wanted using genetic sequencing. So now it's based off of SNPs as opposed to just this cow makes more milk. There's also lots of improvements in automation. There's been improvements in infrastructure and informatics that have helped dairy farmers make better decisions. And so we see that this is an opportunity for us to bring new microbial products based off of sequencing technologies to help farmers make more milk with fewer inputs. And dairy cows are a very good target for microbial products because they contain these massive fermentation tanks inside of them. So you may have heard that cows have 4 stomachs. Apologies if this is an ag group and this is obvious too. I just want to make sure I'm being inclusive. The very first of these stomachs is the rumen, and this is the one that is that massive fermentation tank I'm talking about. And this is like on the scale of 50 to 70 liters inside of a cow devoted to the rumen. And the rumen is what's taking in the plant fibers that the cow is eating, and it's degrading them into sugars. And these microbes are then also fermenting the sugars into short-chain fatty acids, which is what the cow can take up and use for its own energy needs. And so these short-chain fatty acids, they're also called volatile fatty acids in the agricultural world, include acetate, propionate, butyrate. And in fact, propionate is one of the really desirable ones because it is what the cow takes up via the liver and turns into glucose. And so while the past several decades, technology has been trying to improve milk production by targeting the bovine genome, we think that there's a new opportunity to be improving milk production by targeting the genes that are contained inside of the rumen. And these are the microbial genes. So how do we go about designing a novel probiotic for dairy animals? We start in order to improve milk production in particular. And at the start, we are going to collect a whole bunch of different rumen samples, and we want this to work across as many cows as possible. So we're collecting rumen fluid from cows of different breeds. We're collecting rumen fluid samples from cows in different dairying regions of the US, so they're being fed slightly different diets based off of what's available. We're collecting samples from high producers and low producers, and we're taking this collection of rumen samples and sequencing them. We're sequencing the bacterial fraction using 6S ribosomal RNA sequencing. And we're also looking at rumen fungi by sequencing the internal transcribed spacer sequence. We're also doing flow cytometry on these samples in order to get estimates of the total number of bacterial and fungal cells so that we can try and get an estimation, transform what we get, our relative abundance metrics when we do 16S and ITS sequencing, and make an estimation of the absolute abundances using the flow cytometry-based cell counts. We're also running transcriptomics on some of these samples in order to identify which parts of this microbial community were most active. So all of these data then get combined with metadata about the cows that those rumen samples came from, basically how productive were they, and we use them in a network-based analysis. And we're looking for microbes that are not just highly associated with production outcomes, but are also highly connected with other members of the microbial community, because we think those are going to be the microbes that have the best chance of integrating into this ecosystem and having a really robust effect on the animal. So that gives us this list of top target microbes that we would like to develop into a product to help cows make more milk, but we have to then go get them back out of rumen fluid. So we take our rumen samples into these anaerobic chambers, and those that we manage to get into pure culture as an isolate also have to be able to be fermented at scale for a reasonable cost. And then they also have to be preserved so that they can last through the commercial feeding chain, supply chain and feeding on farm, so that they're still alive when they get back into a cow's rumen. And this is easier said than done. A lot of our target microbes that we get into pure culture are vegetative anaerobes, so they really, really hate oxygen. And so they have to be able to survive the process of living in a bag on a farm, just out open to the elements pretty much. They have to be surviving, getting mixed into the feed. You can see in that middle picture on the right-hand side, that's how feed is mixed together. And so our probiotic gets mixed into the feed, into these giant feed wagons, and then they get delivered in front of the cows, where they get eaten up over the next 12 to 24 hours. And so the microbes that we ended up developing that came out of this pipeline that do survive all the way until they get to the cow are these four. This is a product called Galaxis Frontier. I'll be referring to it as Frontier a lot throughout this talk. And it comprises one rumen fungi, that's the Pichiacudria zeviae in the top left, and then 3 rumen bacteria. These microbes encode core rumen functions. This includes degrading plant fibers and like cellulose and hemicellulose. They can also degrade plant starches, not just amylose, but Ruminococcus bovis there in the upper right hand. This is actually a novel species that we first isolated and got to name. It encodes very complex machinery that's required to degrade resistant starch molecules. These microbes can also ferment sugars that are released from these fibers into short-chain fatty acids, including acetate, propionate, and butyrate. And then they're also all producing a vast suite of vitamins, and which are cofactors for the bacteria. And then butyrovibrio fiber solvents down there in the right-hand corner encodes a very cool function called biohydrogenation. This takes plant fats, particularly linoleic acid, which is an unsaturated fatty acid, and it will convert it into vaccinic acid, which is a precursor for conjugated linoleic acid. And so these are core detoxification function in the rumen. So we have tested this product, Galaxis Frontier, in multiple academic studies, as well as on commercial dairies in feed trials. And we're very happy to see that it does what we hoped it would. So what it fundamentally is doing is it's helping cows make more milk more efficiently. On the left side here, this is a diagram that demonstrates the production of milk following calving. And so from left side to the right side, you can see that milk production goes up. And then after peaks around 8 to 10 weeks after calving, milk production slowly tapers off. And we do not have colors here, but what Frontier is fundamentally doing, it is increasing the persistency of the lactation curve so that lactation curve is tapering off slower after peak production. And so they're making more milk over the total duration of their lactation cycle. And if we average it across this whole lactation cycle, these cows are making around 4 pounds more milk every single day. And they're making it more efficiently. So they're not eating as much as you'd expect in order to support that increase in milk production. We've also taken biopsies of mammaries from cows that were on Frontier and compared it to control cows. And we saw that the mammary had changed morphology after being fed Frontier for quite a while. And so what we were seeing was increased number of mammary epithelial cells and milk-producing alveoli. This seems to be supporting these increases in milk production that we saw. And as we started feeding and testing Galaxis Frontier on these commercial dairies that have thousands of cows that we can do in control versus treatment trials, pen-based trials, we started seeing also a reduction in the number of metabolic disorders that occur that are really common around calving. And an overall reduction in early culls. And so we started seeing that it's not just a production effect, but that there are some systemic effects affecting the animal overall. So how do we get these systemic effects and milk production effects from something that stems in the rumen? Changes that are stemming fundamentally in the rumen by feeding these microbial products. So we initially turned to some rumen samples that we collected from academic trials. There are 8 academic trials marked out here in the table in the bottom left, and they were all run pretty similarly. We start by pairing up cows that are most similar in the number of births they've had, where they are in their lactation cycle, and how much milk they were making at the beginning. So we're trying to get cows that are as similar to each other as possible. And then for each pair of matched cows, one gets assigned to a control group where they're just being fed their normal base feed ration. And then the other one gets assigned to the Frontier group, which gets that base ration with a dose of Galaxis Frontier. And so those Frontier cows are getting a dose of Galaxis Frontier every single day. for as little as 16 weeks was the shortest trial, and as many as 40 weeks. And so we were collecting rumen samples during these trials. And this is just looking at the 16S rRNA. So this is just the bacterial fractions of these microbes or of these rumen samples. And the first thing that's really obvious is just how much study specificity exists. And so I told you earlier that animals have a more similar, are on a very similar diet to each other, and therefore we expect their guts to look a lot more similar to each other. Dairy cows are extremely diverse in their rumen. So when we were comparing all of the rumen samples that we had from these 8 different studies, almost half of them, 44.7 of these OTUs were regularly detected in just a single study. And so you can see in that PCA plot there, smack dab in the middle, that these samples were largely clustered just according to where they were collected from. Any changes that occurred due to frontier feeding were a lot more subtle in comparison. And so we used differential abundance analysis in order to identify a subset of microbes that were indicative of frontier feeding. Differential abundance gave us around 200 different OTUs that were differentially abundant in either the control or in the frontier group. Some of them were more highly associated with frontier samples, and some of these 200 OTUs were negatively associated with frontier samples, and we can do a log ratio of those different OTUs. And you can see in the top right-hand corner that the frontier samples in pink were always higher than the control samples in gray. These OTUs, if we turn to the top left, were included a lot of Prevotalaceae family OTUs, indicating a remodeling of this family. And also included a lot of Lactospiraceae family OTUs, although these were mostly in the denominator of this log ratio, which means that they had a closer association with the control samples than the frontier samples. And Lactospiraceae, I think, in rumen ecosystems are very typically associated with plant fiber fermentation, so like cellulose and hemicellulose. And fermentation of these fibers into, or the sugars released from these fibers into acetate and butyrate in particular. So we have a distinguishing signal, but fundamentally these around 200 OTUs were still more clustered by study than really any other feature. You can see this in the network diagrams in the bottom right-hand corner. The study clustering effect is still very dominant. This indicates to us that we're still battling a lot of diversity across these rumen samples, study-specific diversity. And we suspect that what's really happening to drive this consistent milk response in these studies is instead what these microbes are doing and not specifically who they are. But of course, 16S is not going to give us a lot of information about that. So this is where we were really lucky to work with Daicel Arbor and get their an agrogenomics grant through them and Element to do some shotgun metagenomic sequencing. And so we leaned on one of these trials that we ran. This is the University of Florida study. We chose to focus in on samples from this study because the cows were extremely well matched in this study. And these researchers also ended up collecting a lot of additional metadata about the cows throughout the study. In this study, cows showed a 5% increase in milk yield, milk fat and protein yield, and were around 5% more efficient at making that milk as well. So from the total number of cows, we ended up selecting 24 of these matched blocks of cows, so each containing a control animal and a frontier animal. The animals were very well matched at the very beginning with their covariate values. So they were matched by the number of births they have, where they were in their lactational cycle, how much milk they had made in the previous lactational cycle, their body weights. And then when we collected samples at around week 9 or 10, they were still very well matched in the amount of body weight they'd gained, their body energy changes, how much they were eating, but they were making more milk, and so they were more efficient at making that milk. So these are the samples we were using, but we were particularly worried when we were doing shotgun metagenomic sequencing about the amount of plant material that's in these samples. And so we were very worried that when we did shotgun sequencing, that we would be blowing out our microbial signal with a whole bunch of plant matter. And so we subjected it to a couple filters. We initially just did a size fraction filter to eliminate large fragments of plant matter. We took that large fraction that included plant matter, and then we also did a second incubation on it in a second filtering. That was to try and dislodge any microbes that were adherent to the fibers, to the plant fibers. And so then we would pool the two small fractions, one from the incubation to remove plant adherent microbes, as well as the initial filter in the first place, in order to get our microbiome fraction that we extracted DNA from for sequencing. So these are the taxonomic assignments of the microbiome communities in these ruminal samples. I used a tool called Metagenomics. This is an assembly-free based taxonomic classifier. The paper promises to explain upwards of 40% of reads in less characterized environments, such as the Rumen microbiome. And indeed, we got 39.5% of our taxa being taxonomically classified. You can see that most of them are going to be bacteria in green, are assigned to bacterial kingdoms, and then also a small abundance of archaea. You may notice that we have very few eukaryotes found in these samples. Partly this is going to be due to rumen eukaryotes being very poorly defined in databases, including Metagenomics. But I think also this might be reflecting some of our filtering steps as well, that perhaps we were a little too stringent with our filtering. So the bacterial fraction here on the left is showing you kind of what kinds of bacteria we have in here. It's mostly Firmicutes or Bacillota. This database is using old phylum names, but it may be perhaps you're familiar with them still. Includes Firmicutes, Bacteroidetes, Actinobacteria, and Proteobacteria phyla. And then the archaea are actually we're pretty much all part of this a single family, the Methanobacteriaceae family. This includes Methanobrevibacter. These are all methanogens. These are methane-producing microbes that are making methane from hydrogen and carbon dioxide. So Metagenomics ended up calling a total of around 1,000 SGBs. SGB is their unit, their taxonomic unit. It stands for a species level genome bin. And so we're kind of looking at species here with the resolution of around a species, of the species level resolution. What we immediately picked up on is that these samples are, again, very diverse. So if we were looking at just the control samples, only 114 SGBs were shared across pretty much all of the control samples. However, when we looked at the frontier samples, there were about double the number of SGBs that were shared across most of the frontier samples. So this is reflected here in the diagram on the bottom left as well as the table. You can see that on the right-hand side there's a lot more prevalent. There's more SGBs that are shared amongst 21, 22, 23, up to 24 of the frontier ruminal samples that we submitted to shotgun sequencing. Meanwhile, there are more SGBs amongst the control samples that are only just in a handful of those ruminal samples. This is also reflected when we do principal component analysis in the bottom right-hand corner. You can see that the frontier samples are all clustering together in the bottom left-hand corner of this ordination plot, whereas the control samples are much more diffuse. So who are these microbes, these SGBs that are so much more abundant in, or so much more consistent in frontier samples and less so in control samples? They are mostly Bacteroidetes in this, shown in the tree in the bottom low in blue. It includes a clade that is mostly assigned to the Prevotolaceae family. There's another clade that's mostly assigned to Murray-Baculaceae, and then a third clade of Bacteroidetes that is undefined. And I want you to kind of hold on to some of these names because we're going to see them again. But turning back to our principal component analysis, what we could do is we can go in and we can pull out the drivers, the loading variables that are driving the clustering of the frontier samples in that bottom left corner. This was using a phylogenetic-based principal component analysis technique. And so what it did was it was able to group together groups of microbes that were phylogenetically related as part of a clade and define those as loading variables as well. And so in the end, we ended up getting 34 SGBs that were defining these differences, and they collapsed into 5 total clades. Some of them were pointing down towards the cluster in the southwest corner, and others were pointing away from the cluster in the north, towards the north and the east. And we can take the ones that are clustering towards the cluster, or sorry, the loading variables that are pointing towards the cluster, and do a ratio of those versus the ones that are pointing away from the cluster. And again, get this sort of ratio that defines 2 separate groups, the frontier group in blue now versus the control group in red. And these 34 SGBs are also able to be used in a random forest model to pretty well predict what the treatment group is from that rumen sample. So who are these microbes that are in those 5 clades that distinguish treatment groups? In the numerator, we again have 3 clades of Bacteroidetes members. There's a clade of Prevotalaceae, a clade of Muribaculaceae, and a clade that has been so far undefined. And so this is exactly what we saw in the prevalence-based analysis. In the denominator, we see 2 clades of bacteria that are from the family Lachnospiraceae. One was defined as part of the Oribacterium genus, and the other one contains SGVs that are assigned to the Vibrio genus. What's so special about these SGBs was kind of the first question we asked. And so to get at this, I did an enrichment analysis. This enrichment analysis was for different metabolic pathways. And so I looked for the prevalence of metabolic pathways amongst those 34 SGBs that were distinguishing treatment and control samples. And I compared the prevalence of pathways in those SGBs to the prevalence of this pathway amongst all the rumen strains that we saw. And what stood out were a couple reversals in amino acid metabolism. The first reversal we saw was the degradation versus the biosynthesis of arginine. So the numerator taxa, these are the ones that were more closely associated with the cluster of frontier samples, were encoding for the degradation of arginine in making glutamate, turning arginine into glutamate. The denominator taxa were encoding pathways for biosynthesizing arginine through glutamate and through the ornithine, which is one of these urea cycle intermediates. And so what this suggests to us is that the frontier samples are less reliant on these nitrogen scavenging pathways, like the urea cycle, and are instead relying more on glutamate for potentially doing a lot of amino acid interconversions, as well as other central carbon and nitrogen metabolisms. The second metabolic reversal that we saw was the degradation versus the biosynthesis of threonine. So the numerator taxa, again, these are the ones associated with the frontier samples, were encoding for the pathway to biosynthesize threonine, whereas the denominator taxa was encoding for the conversion of threonine into isoleucine. We think that this is relevant because threonine in the rumen can be fermented into propionate. Propionate, again, is one of these short-chain fatty acids that are made in the rumen that are then absorbed by the cow to make energy. And we saw in our rumen samples that concentrations of propionate were increased in frontier cows. And not just in this one study, but actually in all 8 of our academic studies that we have rumen samples from, we saw an increase in propionate concentrations. Meanwhile, isoleucine can be fermented into isobutyric acid. And isobutyrate was not significantly changed in this study or our others, but it was numerically reduced in these samples. So this was very exciting, but in order to test these associations more directly, I did a correlation analysis between the abundance of these SGBs that were differentiating our treatment and control samples with the concentration of various ruminal short-chain fatty acids. In the bottom left-hand panel there, you can see that these numerator taxa that are associated with frontier samples show a lot of positive correlations with propionate concentrations and valerate concentrations. And we think that this is particularly very important for interpreting our animal results because propionate and valerate are used for making glucose in the liver. So what happens is propionate gets picked up through the portal vein, it gets shipped, it gets picked up by the liver in the animal, and then it feeds into the TCA cycle. This is what's called an anaplerotic pathway. It feeds into the TCA cycle, refilling TCA cycle intermediates. And if you recall, gluconeogenesis pulls from the TCA cycle. And so we need to keep replenishing the TCA cycle intermediates in order to continue making glucose. And so this is what the cow really relies on for making glucose, is the constant refilling of the TCA cycle through propionate especially, but also valerate. The other interesting feature about the other fun thing that comes from refilling the TCA cycle is that it allows acetyl-CoA that's being produced by fatty acid breakdown to continue being integrated into the TCA cycle. When the TCA cycle is not able to continue functioning, acetyl-CoA has to find somewhere else to go. And what it typically does is it gets converted into ketone bodies, including beta-hydroxybutyrate. And these ketone bodies, when they build up, leads to a condition called ketosis. And this is one of those big metabolic disorders that happen around calving that we see a reduction in the risk of in our cows being fed Frontier. So this is super cool for us. This was starting to indicate that there are changes happening in the rumen that connect to the animal results that we're seeing. But in the end, this is still just a handful of microbes that are in these rumen samples, right, that we picked out. And so I wanted to zoom out more, and I wanted to look at the full functional potential of these rumen samples, since we have these really nice big metagenomic samples. And so I built out a gene catalog of non-redundant genes from that are encoded in these rumen samples. And I did a differential abundance analysis on all the different genes that were being not expressed. This is just encoded in rumen samples from Frontier cows or control cows. And I took these genes that were being differentially expressed in the two different groups, and I mapped them onto metabolic pathways using KEGG orthology, and to look to see whether or not there were certain pathways that were enriched in each group. Hot off of talking about propionate, the very first thing that I noticed was that the Frontier cows were enriched for this gene, fumarate reductase, which is used to convert fumarate, to reduce fumarate into succinate. The reason that this is particularly relevant is because succinate gets readily converted to propionate in the rumen, which again gets picked up through the portal vein, picked up by the liver, and turned into glucose, which the animal can then use. The other interesting thing about fumarate reductase is that it uses hydrogen to do the reduction reaction. And hydrogen is one of these molecules that can build up in the cow's rumen as she ferments, or as she degrades a whole bunch of plant fibers. And typically, the place for hydrogen to go is into methanogenesis. And so this gets combined with, typically, most abundantly in the rumen, with carbon dioxide, and gets turned into methane, which that is a typo. That should say CH4, not CH2 down there at the bottom. The other thing that I noted when looking at the enrichment of differentially abundant genes in Frontier versus control samples was that Frontier cows were highly enriched for biosynthetic pathways, particularly in cofactor biosynthesis and amino acid biosynthesis. So on the left hand, we're looking at an enrichment in genes involved in folate biosynthesis, in one carbon pools by folate, looking at coenzyme A biosynthesis and porphyrin metabolism, porphyrin biosynthesis, cofactor biosynthesis. We're also looking at enrichments in genes for doing histidine biosynthesis, aromatic amino acid biosynthesis, and branched chain amino acid biosynthesis. On the other hand, control cows were enriched for amino acid uptake, particularly you can see in the upper right-hand corner for amino acid transporters. And to me, this means that these, coupled with fewer biosynthetic genes for amino acids, that these control cows were containing genes that were mostly reliant on uptaking existing amino acids and not biosynthesizing themselves. So what this, I'll describe what's in this diagram. The way that we're interpreting this data is that feeding Galaxis Frontier supports an environment, a ruminal environment, that is really rich in metabolic function. So I told you earlier that the way we chose the strains for Galaxis Frontier partly included strains that had to be very highly interconnected with the rest of the microbes in the rumen ecosystem, in the rumen community. And we think that not by synthesizing cofactors themselves, they're also encouraging an ecosystem that is really equipped to do more cofactor biosynthesis, more amino acid biosynthesis. This is what can be used in order to make more enzymes and use more enzymes in order to do all these core metabolic functions within the rumen. The other upside of amino acid biosynthesis is that once microbes pass out of the rumen and down the digestive tract, say into that fourth stomach, which is a lot more like a monogastric stomach, a lot of these microbes will end up dying, and they'll be releasing their protein for use, but for the animal's actually primary protein source from the diet. And then, last but not least, we see an increase in propionate levels, propionate concentration in these rumens, and we can see that there are correlations with different microbes that are changing in abundance in these samples and the amount of propionate that we're getting. And this propionate we know can be picked up by the cow for glucose production. It might explain why we see such systemic effects, not just in milk production, but also in reduction in coals. So this glucose can be used for host maintenance. It can be used for milk production. It might be altering hormonal regulation, which might be explaining why we see differences in the mammary morphology. And so overall, these changes in the rumen ecosystem are trickling down into the rest of the animal to help her make more milk more efficiently. So I did not do any of the animal work myself. I just want to take a moment to briefly acknowledge all the people who collected ruminal samples for us, did these feed trials that lasted from anywhere from 16 to 40 weeks. These are massive project undertakings. We respect them immensely, and we thank them very much at all these institutes, all these people that helped us. And there's far more people on those teams than I've listed here. These are just the primary people that we interact with regularly. I also want to thank Kayla Kalapa, working with me here at Native Microbials. She's our sequencing manager and does a lot of the DNA, the molecular work, as well as Mallory and Lisa on my team, who are also scientists and the CSO of our company who helped out with everything. And thanks to Daicel, Arbor, and Element for supporting our metagenomic stuff, our metagenomic work with an agrogenomics grant.
All right. Thank you so much, Brooke. That was a great presentation. Brian, thank you for your presentation. We really appreciate it. Thank you to all the audience members who stuck with us through those glitches. We appreciate it. We know we ran a little bit long. We still want to get to some of the question and answers and answer a couple of the live questions that came in. So I'm going to get to those now. Audience members, if you'd like to stick with us, that would be great. If you have to go, we understand it is a little bit over the hour time frame, but I'm going to jump right in it. So these first questions are probably for Brian. This is a question of what type of enrichment is typical for HYBE? Is it really from about 5% to 80%?
It really depends on the, just to double check, you can hear me?
Yes.
Excellent. All right. My AV is now back on track after my talk. But anyway, so for enrichment, it really depends on the amount of endogenous or target compared to the amount of background. So you can get from 5 to 80%. But if your target is something like 0.1%, you might only then get enrichment at 20%. It's still like several hundred or thousand-fold enrichment. As the protocol mentioned, there's a high sensitivity approach where you can then really get that up to like 80%. That's another point of which to check, like, is it worth spending the money and time to do another enrichment to get higher on target, or do you just sequence after the first round? So that's another decision point that is based on what you need to get.
Great. This question is probably for you also, Brian, and I think you answered this earlier on, but I'm going to ask again just so that everybody can hear it. Do you need to isolate them? This was during your presentation as you were talking. Do you need to isolate them and then sequencing?
So it's just straight DNA or RNA extraction from a sample. So it could be from soil, it could be from a cow. There's a study that we helped with where they looked at Bacillus anthracis in Africa from cattle. And so that was really nice that no one had to culture or isolate any anthrax. They could just take the cattle, the full sample DNA, and then enrich the bacteria or virus or whatever they wanted from the sample.
Great. This question is about the PowerPoint slides, actually. We will make these available to you. The recorded webinar will be on the PAG site, so you can come back and watch it. We'll have it streamlined, cleaned up, so you can watch this on demand as well. Next question, Brian, again, I think this came in. How long are the hive cap probes usually, min and max?
So minimum is 60. People will often use that when doing very degraded ancient DNA. The max is 120. And so we recommend anywhere between 120 and 80 usually, depending on if it's very high GC content, you might want to use shorter probes to avoid secondary structure formation. But most times, longer probes will be the most cost-efficient approach.
Sure. This one, either of you can answer or both of you. We'll start off with Brian. How are you approaching variant discovery in complex or poorly characterized microbial systems?
So if you know what you're looking for, let's say SARS or something like, African swine fever virus, the hybridization capture probes can tolerate diversity. So if the probes are overlapping, in this example that we had worked with someone, 80 nucleotide probes every 27 nucleotides. So not only can the probes tolerate 5 to 10% or more divergence in the sample, if there is an insertion deletion, there's always going to be a probe flanking any type of indel or other polymorphism. So it works really, really well for capturing diversity within a set target. If you don't know, like you're trying to identify some bacteria and you don't know what you're trying to get out of it, then you have to do metagenomic or full shotgun sequencing. That's the only unbiased approach, with the caveat that if it's low abundant, you might not be able to really resolve it because, if you do 10 million reads and it's at 1 to 100 million in your sample, you're not going to detect it. But I like to hear Brook's part too.
I could follow up on exactly that from, so there are tools out there for helping profile for strain variation from metagenomic sequencing once you've assembled your mags. InStrain is the one that I can think of off the top of my head. For an application like mine, when I already am tackling that much diversity, the strain level diversity is just an added layer of complexity that, as of today, I haven't dealt with. But I do know that I think in a simpler ecosystem where strain level diversity is matters a lot more, say, than just overall diversity, a tool like InStrain could be very helpful once you have your mags assembled.
Great. This, again, either one of you can answer. Brook, we'll start with you since we started with Brian last time. How are you designing your next-gen sequencing workflows to capture both community composition and functional potential? And if you need me to read that again, I can.
No, so I think ultimately, our pipelines have always subsisted very much on 16S ribosomal RNA sequencing. It is still a tool that is most accessible to people because of its cost effectiveness, because of its low data requirements, and fundamentally, we want to complement all of our data with metagenomics, but it's often very cost prohibitive, as well as the cost related, with the data, not just the storage, but the processing of the data. These are all very expensive. And so our approach that I showed you here was one of using mostly 16S and then complementing a subset of our samples with metagenomics in order to get at functional diversity. But it's going to require cost reductions in order to make metagenomics sampling more accessible to everyone and to make that a more common pipeline.
Great. Brian, any input there?
Well, I totally agree. One of the ways that we, again, context dependent, we would approach it potentially is do shallow sequencing to find the functional genes. You could, if there's regions of interest, like AMR genes, we have a kit for that. And then anything else, like a 16S, you could take the same NGS library and spin it 3 ways. You could do shallow sequencing for metagenomic and functional analysis, and then high level resolution of things that might be of interest, whether it's virulence genes, AMR genes, or others. Because I agree. Unless you're processing data, you realize how much data you get, how much it takes to store, manipulate, upload, download. And if you're doing hundreds or thousands of samples, it becomes really difficult to deeply sequence everything.
Yeah. All right, next question. And we still have some audience members on, so let's just get through a couple of these really quick. Could HibCap process be used for strain species abundance? quantification? If so, how different would the post-processing compared to shotgun metagenomic sequencing like controls or special normalization? You might want to read that one on the questions because I can repeat it, but that was a lot.
Nope, I got it. So HiveCap can definitely maintain the relative abundance in the sample. So you could use it for targeted RNA-seq as well. So there is a paper by Megan Bouldry et al. Where she looked at 16S and compared it to shotgun sequencing, and the community profile is the same. So it's very, very good at not biasing the abundance. The analysis, that's where the trick sometimes comes in. You can use often many of the same metagenomic tools on HiveCap because what you're doing is just taking the same amount of like shotgun reads that you're just enriching for a particular subset. So most times you're fine with that, but there could be some complications depending on if you're like looking at 16S or something specific that might like a high level of similarity among samples, but the relative abundance is maintained. Processing should be straightforward until it's not.
Great. Brooke, any input?
No, not on HiveCap.
All right. All right. Well, here's another question. Thank you, Faith, for sending this in. Hello. Do you have recommended DNA, RNA extraction protocols for short or long read sequencing of different sample types? And is this a service you provide as well? I apologize if this is a repeat question. I missed some of the presentation earlier. So it's not a repeat question really, but I'll let you guys take this.
So I would say that is, yes, we do have recommendations. I don't know them off the top of my head, but in our services, we'll try to, we offer extractions and we definitely have different protocols for like insect extraction versus wheat extraction, for DNA and RNA. And because you want to maintain high molecular weight for long insert, we also have different, protocols or maybe even steps, to make sure you have the correct inputs going in. And so if you want to reach out, I don't know what those are, but I'm sure we can provide you with, what we use.
Yep. And Faith, we can definitely have somebody contact you via email and answer some of your questions that way also.
Yeah,
so
definitely. I'll put you in touch with the person who knows more than me.
All right, here we go. Can you use same cow and just add treatment in a consecutive location cycle or lactation cycle that is run experiment in 2 lactation cycles, one with treatment and one control? So that's probably for you, Brooke.
Yeah.
I hope.
This would be super fantastic if we could. The reason we cannot is unexpectedly, the microbiome changes a lot from 1 week to the next. It changes from one lactation to the next. And actually, the biggest thing that happens is an academic institute usually can't store enough feed to feed the same animal, the exact same feed for that amount of time. And that's really one of the defining features here. So if we ran it over 2 consecutive lactation cycles and used the same animal as its own control, it might be a little bit cost prohibitive versus just doing something for 16 weeks. But B, it's probably going to be changing in that time in ways that we can then not account for, are confounding basically your treatment effect. So the way that we get around this is by finding cows that are as close to each other as possible and then distributing them into control and treatment groups.
Great. What other analysis tools did you consider besides I'm so sorry if I pronounced this wrong. Flan, yes, Metaflan. And do you know what other sample types besides rumen that Metaflan would be appropriate to use with?
Right. I'll answer the second part first. So Metaflan is a really good tool for things that are really well described in particular. It's a tool that's based off of using marker genes. And so this is how it gets away without needing assemblies. It doesn't do full alignments. It just is mapping reads to specific marker genes that define these species level genome bins. So any community that's particularly well described, this is especially going to be like human gut samples. It's going to be really good at those. What other tools did I consider beyond using Metaflan? So I also did do full assemblies of these. I assembled them into metagenome assembled mags. I mapped them. I used taxonomic assignment with GTDB. I don't actually know what that stands for off the top of my head. Genome Taxonomic Database, maybe. And so there are these, I would break them into assembly-free tools like Metaflon, and I'm not familiar with many others, and then assembly-based tools for taxonomic assignment, of which there are many that I know Brian briefly touched on. Kraken, GTDB is one. And these are based off of doing an assembly and then binning your reads into metagenome assembled genomes, and then doing taxonomic assignment of those MAGs. The reason I didn't present that here is because I think it is limited and it is biased according to how abundant those species are. There's also a number of errors that can occur in assembly. I felt that the Metaflon data was actually going to be a better representation of the overall communities. And since I wasn't specifically targeting any single MAG or strain variation within a MAG, Metaflon felt like the right tool.
Great. All right, when you're doing 16S-based metagenomics and high inhibitor matrices like rumen or soil, what best practices do you recommend to reduce amplification bias and overrepresentation of certain taxa? Are there specific primer sets or library prep tweaks you found work best?
So I think this is for 16S and ITS. What I can say is every single system actually seems to prefer slightly different regions of the 16S sequence, and I'm much more familiar with 16S than I am with ITS. For example, what we are using for cow rumen samples is the V1 to V3 region, but we have found that for other samples, the V4 might be better. It does seem to be, as you mentioned, related to potentially some bias there, but also just where we can get resolution amongst the strains that are most abundant in those ecosystems. My recommendation for your own purposes is to do a lot of literature searches to see what the community has pretty much come to a consensus on for which specific primer pairs, because it does seem to be a context and environment specific choice.
Great. Thank you. All right, audience, we're still here. We still have some audience. We only have 2 questions left. If you could hang out with us for about 5 more minutes, let's get through these. Any recommendations over host depletion methods, kits for enhancing bacterial sequencing in MMS?
MMS, microbial metagenomic sequencing, maybe?
I would say so.
For host depletion methods for enhancing bacteria, I guess a lot of these QC pipelines for metagenomics, in my experience, have a step for host depletion. I was using the Huttenhauer's biobakery pipeline for most of my analysis. I input a bovine genome to make sure that host reads were removed. For the rumen ecosystem, there's not a ton of contamination from the host. I was more worried about, say, plant matter contamination. My filtering did a very good job when I tried to deplete reads. And there's a lot of this can be handled by informatics. For kits, actually, Brian, do you have advice for kits?
There's one I'd used. It's the, I think, NEB microbe enrich. I can't really remember the name of it. I'd used it on pig samples from tonsils because I was having really trouble getting at some of the microbial community. And it worked pretty well. I can't remember the exact name, but it was NEB, and it basically works by depleting anything that's methylated, which should be the host, not the bacteria.
Great.
And that's someone I know. There's not a lot of depletion options out there, unfortunately.
Yeah.
All right, we're going to get one more question in, and then we're going to have to call it quits. Here we go. What are the standard databases you use for 16S and ITS1 metagenomics representatively?
This also is a choice that is environment dependent, unfortunately. So what we've realized is that, for example, the Green Genes database, which is very commonly used, I don't actually know if it is ITS. I can't remember the ITS. I don't work with a ton of fungal data myself. But for 16S, the Green Genes database is not very good for rumen samples. So we use Silva, but I know that there have been attempts to make rumen-specific databases. They are not particularly publicly available. So you have to see which one gives the best hits, best descriptions, closest matches to your particular environment.
Great. Brian?
Oh, I'm sorry. That was, yes, that's right. Okay, go ahead, Brian.
Brian, for
I totally agree.
something?
I don't know much about fungal either. Silva has comprehensive database, non-redundant database. Green Genes 2 is great if you're using Chime. It's basically patched and linked into there. Maybe overrepresented on V4, but if you're doing straight amplicon, it's good. I think if you're doing more metagenomics, Silva might the full-length non-redundant database might be a good one to try.
Well, and actually through Chime, you can. I'm fairly sure you can train some of your classifiers on these different databases, given the certain regions you have. So you can always use a Silva database even with Chime, which is good.
Nice.
Great. Thank you both. Really appreciate it. I love this kind of chat at the end and the question and answer portion of it. It really does make it a lot more real, and we appreciate it. Brian, thank you for your presentation again. Brooke, thank you. We'd also like to thank Daicel Arbor Biosciences for their support and supporting this webinar, support of PAG webinar series, and all of you for joining us today. We had a lot of people stay on, 25 minutes later, so thank you so much. The on-demand webinar will be available on the PAG website within 48 hours. Usually it might be a couple more, another day to edit this one a little bit. We thank you, audience, for sticking with us through these issues that we were having, and we look forward to your attendance in future PAG webinars. Thank you.
Thank you.
Thank you.